Good morning. Good morning. We are a cozier crew than was anticipated. A couple of panelists had some unforeseen activities that they had to tend to, and they send their regrets. But nonetheless, we're going to have an exciting conversation here. Thank you, Paolo, for the wonderful introduction of the final day of Meeting on the Med. It's kind of nice to be speaking on the final day and sort of processing all that's happened in the conference so far. I think a couple of themes that have been spoken about, sort of big elephants in the room, a lot of uncertainty in the world, and specifically in our sector, but I think there were so many successes last year, the number of products that were approved, new technologies that are now making their way to patients, and I think scientists got to science. The world has to go on. People are still going to be diagnosed with diseases. We're still going to be curious about finding new ways to address those. And I think we just keep moving forward. It seems to be the theme of sort of all the sort of panel conversations as well as private conversations that people have been having is we just have to keep going and just assume that the world will just follow suit. So let's lead the world that way and be an example. I also can't help but think about the fact that it's 5 years since COVID. When 5 years ago, this meeting, we all had flights to Barcelona that were canceled. And now we're here, so it's still, that was not that long ago. But yet here we are, we're together, we're gonna have great conversation thinking about the future of cancer. So I'm Jessica Carmen, I'll be moderating this session. I'm the Chief Commercial Officer of RoslinCT. We're a cell therapy CDMO with locations in Scotland and Massachusetts, US. And let the panelists introduce themselves.
Hey, I'm Reagan Jarvis. I'm the CEO at Anocca. We're a Swedish-based T-cell therapy biotech focused on TCR T-cell therapies in oncology, first up. Company's built around a unique cell biology platform for target mapping, TCR generation, characterization of on and off target activity, and then directed evolution of TCR function. Basically, we program human cells as analytes and compile very standardized cell-based assays as a discovery mode. Very deep pipeline. First products going into clinic with in-house GMP capability. CTA here in Europe was cleared just a few weeks ago, so we'll start initiating sites in the coming weeks and start screening patients before summer. So that's a very focused trial in pancreatic ductal adenocarcinoma. I often say we'll pick the easy indications first, but there'll be a fun trial, and a lot more to come.
Good morning, everyone. I'm Kristin Yarema. I'm the CEO of Poseida Therapeutics. Poseida, I now get to say Poseida is a member of the Roche group because we were acquired by Roche at the start of this year, back in January. And Poseida, we've got a foot in both camps here. We do allogeneic cell therapy as well as non-viral gene therapy and genetic medicines. We have an underpinning toolkit, molecular toolkit, that is based on transposon technology and a proprietary gene editor we have called Cas-CLOVER. And we use those tools ex vivo, manipulating cells ex vivo for the allogeneic CAR T, or in vivo, although we're still preclinical on the gene therapy side, but in vivo to do genetic medicines and whole gene insertion using non-viral techniques. We are clinical stage with our allo CAR T portfolio. We have had 3 programs concurrently in the clinic, all phase 1. Our lead program is now in phase 1B. That is an allogeneic program, PBCMA Allo 1 for multiple myeloma. We also have a dual CAR T that carries 2 full-length CARs, which is something we can do using the transposon. For CD19, CD20, that is in non-Hodgkin's lymphoma. We have also cleared INDs for both lupus and for multiple sclerosis with that program, so we're excited about that. And we've had a solid tumor program as well, and that continues to be an area of interest for us. So really thrilled to be here. I think that we have reasons for caution, reasons for concern, as you alluded to, Jessica. But, I was just in a conversation last night. I flew up to Basel to meet with the CEO of Roche Pharma. First time I've been here since the acquisition. And Teresa Graham, her perspective was, look, these technologies are absolutely transformative. They will be meaningful for, many thousands of patients over time. And, there may be a time horizon associated with that, but science and medicine win out in the end. And I think that's, I know we all appreciate that because we're here and we're the cell and gene community. But, it was honestly, it was reassuring to hear that sentiment from, kind of our big pharma colleagues who work across all kinds of different modalities, right? So, we're keep calm and carry on, I guess.
Indeed. I think there's so many directions we could take this conversation, thinking about sort of the future of cancer. I think one of the topics we discussed in our prep call, which I think is maybe worth sort of starting right off the bat, speaking of sort of killing the big elephants in the room, is the pivot of a lot of cell therapies toward autoimmunity. And I think a lot of us have seen the meme that's been circulating on LinkedIn of like the mother catching the beautiful baby labeled autoimmune and the skeleton labeled oncology at the bottom of the ocean, with this notion that autoimmunity is going to, be the new favorite for cell therapy and oncology is just going to languish. And I think that's a pretty bleak picture to paint for oncology. And so, let's talk about that. Is it true? So I guess let's go there. So first of all, is that true? Do we think that oncology and innovations geared toward oncology are going to die in the presence of the new favorite?
Well, I certainly don't. I'm happy to start in on that one. And I feel calibrated in saying that. Our own company is now in both spaces. Poseida started, defined itself as an oncology company, and we have now moved into autoimmune disease as well. But, that is not diminishing our focus. We still haven't cured cancer, though we are coming closer than we ever have with cell therapies. So until we crack that problem, I think there will continue to be interest. What I think is great about the autoimmune opportunity is, first of all, really expanding the horizons for where cell and potentially gene therapies can play, right? So just really opening the aperture on that. And these are very large markets with lots of patients, chronic symptomatic patients with high unmet need. And, frankly, the more value creation, the more patients that can be treated with transformative medicines, the more exciting a space it is for capital investment and partnering and all of those kinds of activities. So I don't see autoimmune, relegating oncology to being the skeleton. I don't see that happening. I think it's, another child that's entered the family, and we can have many healthy babies and all,
I like that.
Yeah, well, I obviously don't think it's going to be a huge challenge moving forward in oncology for T-cell therapies as well. We're just about to launch a phase 1, in fact, that's our conviction. CAR Ts have shown that they can deplete B cells really well in the oncology setting. It's a good home for them in the autoimmune setting for that B cell reset that everyone's talking about. So, it's a good new application for the modality. Cancer where CAR T cell therapies have struggled in the solid tumor setting is targets. What are the genuinely tumor selective targets that you can leverage with a CAR T targeting modality, and that's where our strong view is TCRs come in as the targeting receptors, which open up this brand new, highly tumor selective target space, with the caveat that it's highly segmented across the patient population, so you need to bring more products to bear on those populations. And the biology is just a lot more complex. CAR T was a beautiful workaround of T cell targeting complexity. It's taking the Fab domains off antibodies and fusing them with some T cell signaling domains. Great. But it was a workaround for the inherent complexity of T cell targets, HLA restricted peptides, and the targeting receptors. So it's incumbent on us and the field to try and reduce that complexity, systemize it, and work through the target space. We saw Adaptimmune get their TCR-T product approved last year. The modality works. Now it's about scaling across that target space in a time and capital efficient manner. I think that's where we're at.
I think that's a really great point that you're raising. So, it may be in some ways not that autoimmune is taking over from oncology, but we're in this kind of apparent gap space between the low-hanging fruit, if you will, in the heme malignancies, where we've made great progress, and an even more vast space in solid tumors, but we just haven't been quite there yet. And certainly target selection, dealing with multiple target, the gating problem, and not gates, or where we haven't really quite seen that realized yet, is an important aspect. But I think we're making progress beyond targeting as well. So how do we address the tumor microenvironment? So through armoring and cloaking strategies. In our experience, so as I said, we focus on allogeneic, and what we have come to appreciate is actually the lymphodepletion regimen is a big part of the equation. We study that really quite systematically and found that not only does the lymphodepletion regimen matter even in heme malignancies, but in the solid tumor space, you probably just generally need more. You may want to look at different combinations, different regimens beyond Flu/Cy, for example. And so there's just an awful lot still to learn, that complexity that you talked about for the solid tumor space. But that doesn't mean that people aren't interested. It doesn't mean that that's not an investable thesis either. It's just people are waiting to hear what science you're bringing, and I can say our new colleagues at Roche and Genentech are incredibly excited about the solid tumor opportunity. So we will continue that work for sure.
So I think, to me, well, I guess for those who know me, know that I look at the world in manufacturing goggles. So I always think about how are we going to make them, and make them at scale. And I think, you mentioned sort of heme as low-hanging fruit, and I think it's also been helpful that that's a smaller scale, and addressing the number of patients with the heme indication has worked, with the technology as it is. Autoimmunity brings a whole another level. The patient numbers are sort of mind-boggling of, sort of current state of cell therapy manufacturing. And I think that is, as we've all seen the exhibition hall here, right, there's a lot of companies now that are developing technologies that are going to really help us bring cell therapies up to scale. I think addressing autoimmunity is going to be the kick required to meet scale, so that when the technology for solid tumors comes, we'll be there. So I see the pivot to autoimmunity as an accelerator for addressing solid tumor from a manufacturing perspective.
Well, I think that's absolutely fair, right? I mean, just more scale, more investment, more talent coming into the space. It drives costs of goods down, drives reliability up. It's like every technology ever. It starts expensive, and then you drive down costs and drive up reliability with time. So, and it will take time.
And you know, when we think about cost, a lot of that is just straight production cost and COGS, manufacturing costs. But you know, we have a lot of other costs involved in delivering care and the total cost of care to a patient. So I think something else that we will really learn, we'll have to learn from cell therapy moving into the autoimmune space is going to be setting of care. You know, these tend to be very large practices. A rheumatology practice can have up to 1,000 patients in it. And the patients are dispersed. They're used to being treated in an outpatient community sort of setting. And I think we're going to have to build those muscles and make sure that we can deliver care in those settings as well. And you know, that will also, I think, further serve the solid tumor opportunity.
Yeah. I think that makes sense. So another sort of big hot topic in the field, and that has been discussed in a few panels, is the impact of artificial intelligence on cell therapy product development. So I should say also, actually, it's interesting that we're all here cell therapy focused, you know, because I think when it comes to oncology in cell and gene space, it's really more heavily skewed towards cell therapy. So I guess I don't want any of our gene therapy colleagues to feel like they're being left out of the conversation, but I guess by nature of who's here, we're going to focus sort of on cell therapy. And so, but you know, what are some roles that artificial intelligence could play in cell therapy development? And you know, is it, you know, will it actually help us bring products to patients faster, or will it help us scale faster? You know, what can artificial intelligence do?
So, you know, anytime we talk about artificial intelligence, you know, the first question I always have is, you know, what flavor of artificial intelligence are we talking about, right? Because it's exciting, but it's also such a buzzword. And I joke about this at home with my husband all the time, because he's doing an organoid, you know, AI-enabled organoid company, and I'm like, what are you doing, really, right? So, so I can speak to where I've seen predictive analytics in our work be very powerful, and that has actually been in patient identification for rare diseases, including rare tumors. So I think the same would be true, you know, for gene therapy. I think your point is right. We're talking about cancer, and that's right now more cell therapy focused. But you know, at my previous company, Atara Therapeutics, we were using effectively real-time claims data, unadjudicated claims data, and mining that using predictive analytics to really anticipate where patients might be. And that was really important, because the particular tumor we were working on had patients were progressing very rapidly, and the mortality was very high. So the patients were both rare, but didn't have time to wait for therapy. And so, you know, that was an approach that, you know, I would not have thought of. Somebody brought it to me and suggested it, and we looked into it, and first in the trial, and then in commercialization. And, you know, I will say that was incredibly powerful, and that would not have been possible without the kind of data science capabilities that we have today. And I think that's just the tip of the iceberg. So, you know, I think others are you know, probably going to speak more about target selection, or you know, other things we can do with machine learning, for example. But I think maybe, Reagan.
No, I completely agree with the clinical development, patient selection. I think one of the challenges there, if you're building actual ML models in that space, not just the predictive analytics, then you have to worry about, where's the hypothesis being tested? How long does that take to test? How do you feed back to make better models? Because that's how ML works, is iteration, essentially. And some of those hypotheses will be billion-dollar projects to test, essentially. And so I don't think there's a huge amount of potential in refining models beyond the more classical data sciences, predictive analytics that Kristin was speaking to. In terms of looking at the different flavors of AI and machine learning, I tend to look at the level of complexity across the biology. And at this stage, you've seen in the small molecule and biologic space, AlphaFold type models being quite valuable for predicting potential novel molecules that can be rapidly tested in reliable assays. And that's the thing, it's iterative. They refine their models. They're $10,000, $100,000 assays. They're quickly fed back. You can build your model better, and that's great. That's a great application. In the cell therapy space where we work, there's a lot of complexity in a cell. To say we're going to model cell biology, I think is beyond the data sets that we currently command. That's as simple as that. We don't have the data to feed into it. Generally speaking, we don't have the assay systems to test hypotheses rapidly with, let's say, native and near-native sequence inputs, which you require to refine these models for real-world outputs. So we have a very, very firm view inside the company. We have a big software capability at the company, which is more about capturing and transforming, visualizing the very complex cell biology data sets we generate, but we've always had an eye on where the ML applications come from. And one of the unique things about the HLA peptide targets and TCRs at the center of T-cell biology is that it's actually a reducible problem if you can manipulate the biologic and genetic parameter space in a systematic way. And we have begun to build models in this area, both about target prediction, which is a unusually tractable problem because the peptides match the HLAs in a somewhat formulaic manner. I think the biggest challenge there is reaching through to what we refer to as immunogenicity. Just because you've got a target presented doesn't mean that there are native T-cell receptors or available engineered T-cell receptors that can access that target. And that's both a read on the biophysics, quite frankly, so AlphaFold will be useful there, but it's more of a read on thymic biology. It's a very sophisticated system where all our self-peptides are negatively selected in the thymus, and that trims the potential diversity of that repertoire. So that's a very long answer to say you need to have real-world biology data in high parameter space, and sometimes unique parameter space, to actually build these models, refine these models so they're useful. And the use cases here are better target selection, better targeting receptor generation, and potentially downstream of that, being able to manipulate the cell products themselves there to behave in a more desirable manner.
So would you say it would enable us to get to like a faster to fail a candidate, in order to get to the real candidates and sort of weed through the maybes and get to the sort of most likelies?
Yeah, I mean, that's it. I mean, you need to fail these in very early discovery. There's such a lot of target space and such a lot of receptor space that your darlings need to be killed almost immediately, and that's really where the fast fail concept comes through. And today we do that with biology. We build these cell-based assays to do this in an empirical manner. And for cell therapies, the number of therapies we need to generate today don't really need the ML leg up. You just do the work empirically. But in future, as you look at the available target space, whether it's personalized, individualized vaccines, combos with cell therapies, which I think is a very strong play moving forward, you need to generate the volume of these precision therapies that you will need those machine learning leg ups to cut down the wet lab work that you're going to do empirically. And I think that's where the playoff, it's not AI or biology, you need to synthesize the two. That's the only way to move forward here.
Yeah, we look at the personalized, the complexity of the personalized angle from upstream. So because we do allogeneic cell therapy, our starting material is individual healthy donors. But you want consistency of the product, and you want a large batch to be able to be used and to be safe and effective in many patients. So really understanding, what goes into quality donor selection, how much is really attributes of an individual donor versus attributes of your process, versus, minor fluctuations in conditions. There's an awful lot to process, and we will be looking at AI applications, in that particular space. So
yeah, that makes sense. I mean, I think thinking about donor selection and target selection, so the sort of identifying the right patient pool and the right candidates and the right product to develop. I think that all makes sense. It would be nice if we would see shorter time to develop, product development timelines shorten as a result of the ability to introduce AI applications, and have us be able to get to clinical trials faster with candidates. That would be really exciting to see, I think. So as you both were speaking, something that sort of popped into my head, which we didn't actually discuss in our prep call because this is sort of a new hot off the presses topic, the announcement last week that the FDA is going to make steps to move away from animal testing. And there's going to be an AI component there, or organoids. Maybe there's a role there. Maybe your fields will merge in your family.
Yeah.
So I guess what are your, and this is sort of so new, and I think we're all still trying to understand and sort of process ourselves. But what do you think are the implications of that, or what was your feeling when you saw that news? Did you think, oh great, yay, like no more animal studies, those are expensive and timely, or was it, oh, I'm really concerned that we're not going to have a good sort of surrogate in a biological model before we go into patients? What are your gut feelings?
Well, I thought it was great, personally. And it's particular in the TCR field. This has actually been fairly long-standing from the FDA. Adaptimmune, Amatics, Immunocore have all submitted INDs without animal data, because there was a recognition that the toxicity packages for TCR-based therapies are just not relevant in animal models because they don't have human TCRs, they don't have human HLAs, they don't have a human genome. There's nothing relevant there to be testing toxicity against. And this is one of the similar interactions with the European regulators and our regulatory scientific advice and CTA submissions. So I think it's great in a very constrained field like this, where it makes perfect sense. I won't speak more broadly because I'm not an expert.
No, I completely concur with that. And so my husband and I both happened to be home when that came out, and kind of, it popped up on the news feeds at the same time. And of course, it's a fantastic goal, right? We would all love to work without animals. There's the ethics of, working with the animals, there's the expense, there's, the consistency, there's the fact that the animal models are, not always that great. So a very, very worthy goal. I think the challenge is going to be in many fields, less relevant for ours, but I think the perspective from people who are really working in that right now is that's a big ask, a big statement from FDA, and a big assumption about the state of the field that's probably many paces beyond where it actually is right now. So a great goal, but if that's the expectation overnight, probably a lot of therapeutic developers, and depending on what the modality is and what is the disease state, that's going to be a challenge.
Maybe just to add, it's definitely modality dependent, right? And maybe one thing to say, whether it's your preclinical model was cell-based, organoid-based, AI-based, you need to be in a position to say this is a genuine model that's genuinely testing the parameters that are important. And we're not setting things up to see what we want to see. I think that's one of the challenges. It's also one of the challenges in animal models, of course, but people need to be honest about that preclin and fail fast. Not talking toxicity, I think talking efficacy in particular. But toxicity is a question, I think outside the cell and gene space very specifically that needs to be dealt with carefully.
Yeah, it's interesting, and I don't think that the elimination of animal studies would necessarily accelerate timelines, because I think there's probably more data there's still going to need to be a good body of data to be able to effectively demonstrate that what we're developing is safe to go into a human, or at least worthy of testing the safety of.
Yeah, it depends, right? So again, maybe not so much for our field, but for small molecules, if you could eliminate 2-year carcinogenicity studies, I mean, that's a huge bottleneck, huge delay, part of your cycle time with that kind of thing. But it's, I think one of the great features of cell and gene therapy is, we have trade-off, we have large upfront capital expenses. We have, we're at the bleeding edge of the science and technology, and we're doing, things that maybe are in some ways less validated. So we have other things to contend with, but questions of toxicity, questions of reprotox or carcinogenicity. At least we're spared those things.
Yeah, I think that's right. All right, we had a few more topics we discussed. So what new technologies do you see coming? Have you heard of, or have you sort of thought of, or that you would like to see? Do you think that could be sort of entering the market and maybe address some of the unmet needs in oncology?
I'll jump in and say TCR targeting and cell therapies, of course. So, I mean, this is accessing target space. Like I said, a first approved product last year, and I think this is demonstration that that targeting modality works in a cell therapy context, and now it's about scaling out across that extremely rich but extremely segmented target space. And a lot of enabling technologies required to make that happen, not just discovery like is the core of our company. But manufacturing-related technologies are extremely important. And that's not just about the biotech, it's not just about the reagents, it's also about the engineering solutions, which is becoming more and more of a focus. And I mean real-world engineering, automation, systemization of the manufacturing process, and keeping your eye on both of developments in both areas is super important, because you can engineer yourself into a corner in both respects. You don't want to over-automate too early. You don't want to over-engineer your biotech too early. It reduces flexibility. And there's a sort of unique perspective that we take in the TCR-T space, is that there are so many products to build. Our pipeline, terrifyingly, has over 40 asset candidates already, but it does give us the opportunity to innovate as we move through that very deep pipeline. And so we've built internal process development and what we call a tech development team as well, that screens new biotechs and new instrumentation to implement in the process development team. So it's not about saying we're going to do major manufacturing changes in pivotal trials, but we have the opportunity to keep innovating on a continuous improvement basis through our pipeline. And that's, I think, very important in a space that's very rich in technology. A lot of innovation already there needs to be synthesized and standardized in the correct way to make these products fly in a cost-effective, consistent manner.
Yeah, I absolutely agree with all of that. I think TCRs are, very exciting. I think we're going to see a lot more there. That offers the opportunity also to open up just so many more targets. We've talked about solid tumors already. I think we'll start to see mixing and matching of these things. So, you know, depending on how good your transgene insertion technology is in your editing, you know, can you mix and match multiple CARs and TCRs, for example, you know, to, again, try to address some of that complexity that we touched on earlier. So I think we'll see more there. You know, certainly we're, you know, team allogeneic, and, you know, I do personally believe that that is going to be an important part of the answer for scaling out these therapies to meet the volumes of patients who really deserve to benefit from the transformational promise of these therapies. I think we are at a tipping point for allogeneic technology. It has been a challenge for many, that is absolutely true, but I think we are collectively turning the corner, and we're going to see some successes in the near future and build upon those. And that will be another way, not just to produce at greater volumes, but to really drive down the cost and try to open up some of the access and commercialization questions and barriers that also have been, you know, plaguing us as a sector a little bit. But there are, you know, there are many other things I think coming. I think we all are carefully watching and have high hopes for the in vivo CAR T technologies, which are not technically a cell therapy, but we, you know.
There's our gene therapy colleagues. There they are.
We kind of group them together. You know, tough to get that right, but game changing if you know, if people can find the right combinations of, you know, delivery vehicles with nanoparticles or whatever that can really target the right types of cells and get, you know, high enough efficiency there. So I think we'll look at that, and I think we'll also continue to see extension into new disease states. So we've talked about oncology and, you know, our new adjacency, autoimmune, which is kind of all in the immune system. But I think we may start to see people, you know, using the same approaches, but going into new and different disease states as well.
I think that makes sense. So sort of keeping on the theme of sort of new technologies, there's been a bit of a renaissance for ADCs in the last few years. We've seen quite a few ADCs become approved and sort of rise to the top, sort of taking, you know, getting closer to frontline. Of course, nothing's going to supplant chemo. Well, maybe it will. Actually, maybe that's another topic in and of itself. Well, how about that? Actually, do you think that that any technology could supplant chemo as frontline for cancer?
Yeah, I mean, it depends, I think, to some extent on what kind of tumor are you talking about? How bulky is it? You know, do you need, can you, let's just say, you know, you want to use a cell therapy to do that. Can you debulk the tumor enough without having, you know, a cytokine storm, CRS, so that you're in the therapeutic window and you're threading, you know, you're balancing the efficacy, because you need to get to a very deep remission, right? Or solid tumor, as much tumor shrinkage as you can get, but that's the goal, right? So, you know, can you do that while maintaining acceptable safety and tolerability? That's what you need to do. And if you have a low tumor burden, I think we will see that. If you have a really high tumor burden, that's where some kind of adjunctive, you know, therapy, chemo would be the obvious choice, is potentially helpful. So I think we'll see the rise of a lot of kind of consolidation type regimens, right? Debulk and then consolidate with a CAR T instead of a transplant, which would, you know, be today's paradigm.
I think that's dead right. I mean, chemo's not going away. There are a lot of incredibly useful clinical tools there, and the physicians know how to use them, and they're part of standard of care for a very good reason, is that patients do benefit. And maybe just a perspective on our first clinical trial program, it's very much considering that in PDAC, in pancreatic ductal adenocarcinoma, where fulleronox is standard of care upfront chemotherapy, and a lot of patients actually benefit. That's why it's standard of care. But it's a very tough chemo to tolerate, and go 3-4 cycles, and basically you have to stop doing it, and patients progress. So our clinical design is basically enrolling at diagnosis, taking the leukopak, autologous gene-edited manufacturing. They go on standard of care, 16 weeks standard, for a few cycles. They wash out, and we've manufactured in the meantime, and we're trying to consolidate benefit from the chemo as a second-line therapy, sort of stomp that those tumor masses once they've started to be gnarled up a little bit by that chemo.
Did you say you take the leukopak before they start?
Yeah.
So that's I think one of the biggest challenges is sort of if you can envision a world where the CAR T could come before chemo, because it's we know these patients that have had chemo, and what other second, third line when chemo gets to be 20th line therapy or a CAR T, these cells are so unhealthy from all the battering they've had, not just from the disease itself, but then from all the library of treatments the patient has had. So actually to be able to take the leukopak before the patient sees the therapeutic agent is actually really compelling.
But it really is the view on trying to integrate with standard of care in a very focused way. I think one of the lessons that the TCR-T field should take from the CAR-T field is the focus of the clinical programs. They were very well defined sub-indications of these B-cell malignancies that you could map a TPP, have an approvable product on a let's say a timeline, but at least a view of how you would actually seek approval. And in the TCR-T space, it's been this big basket trials, searching for signals, and we'll double down later. And I think that's wrong. I mean, CAR-T was very successful very quickly because of the focus. And solid tumor setting is a bit more complex, so you just need to be very cognizant of current standard of care and how you're going to integrate in a very focused way.
Yeah. Well, and then actually getting to sort of the ADC and the radio pharma products. So is there a role there too? Because I think, I think those products tend to do a really good job of modulating the tumor microenvironment, maybe more than the actual killing of the tumor. And so I think there could be, you can envision some combinations or maybe some sequential treatments where you're really, because we know it's not just the tumor itself that's the problem, it's the environment and the ability to get in, to really get in there and get all of it.
Yeah, no, absolutely. It's a renaissance, like you said, of ADC and radio, pharmaceuticals. It's like a flashback to, 15, 20 years ago. And those products were probably ahead of their time in that day and age. We didn't really know how to develop them or use them, and now things are kind of back, I suppose. But it is a mixed experience. You do get different toxicities, particularly with the ADCs, that in some tumors are quite troubling, right? So we're working right now in myeloma, and the ocular toxicity from an ADC is not something that the patients enjoy, and it's something that actually the hematologists, the hem-oncs, really don't want to deal with, and they really don't want to call in their ophthalmologist colleagues for a consult. So you get these, you have more factors that you're kind of potentially having to deal with. But that said, in some cases they've been spectacularly successful, and the possibility for combinations of these different sorts of modalities is something I think we'll start to see some steps into in the future on the horizon. One of the things that that Roche had interest in Poseida, we had a collaboration for a couple of years before we were acquired, And always in that collaboration concept was the idea of CAR T combinations with other stuff. But if you're going to do that, you need to have agents that are effective, but also intrinsically safe enough, or have an amenable tolerability profile, so that you really can do combinations or sequences.
Maybe just generally on the bispecifics, ADCs, radiopharm. I mean, the ADCs, radiopharm, the chemistry is getting better, the isotopes getting better, the supply chain is getting better. That's all great. It does come down to targets and indications, and I completely agree about combo therapy approaches. I mean, multiple targeted agents are likely to be extremely beneficial in some cancer settings, some indication settings, but that's a long road. You need to demonstrate safety and not necessarily synergism, but at least additivity in the therapeutic approach, and that is a lot of patients through, and they're complex trials. So I agree that there's a big potential there, not just ADCs and radioligand therapies, but other targeted agents as well, to try and consolidate, build adjuvant, neoadjuvant type of thinking around cell and gene therapies, because you have this in the cell and gene space. You have this our tagline for as a company is engineered immunity. This is an existing powerful mechanism that you're trying to harness, but in most instances, that's not going to be enough to completely clear a tumor all by itself. And I think everyone recognizes that these aren't magic bullets in the cell and gene space. There's a lot of work and a lot of combos that need to come through moving forward. But I think everyone's aware of that, and most people are thinking very hard about it.
All right, well, we're just about out of time. I think this has been a very good conversation. I think it feels like there's a big, bright future for cell therapy and maybe gene therapy in oncology. And thank you all for joining us on the last day of Meeting on the Med. We'll all be here if you have any further questions or want to talk more about cancer. Okay,
Thanks.
Thank you.